I am Renee Romero, the Director of the Washoe County Sheriff's Office Forensic Science Division, otherwise known as the Crime Lab. Our Crime Lab is one of only 2 accredited crime labs in the state of Nevada. The lab is accredited by the American Society of Crime Lab Directors Laboratory Accreditation Board, an independent organization that establishes minimum operating standards and monitors compliance with these standards. We provide forensic services to local, state, and federal agencies in 13 counties across Nevada. The Crime Lab is a full-service forensic laboratory staffed by over 30 individuals in sworn and civilian capacities. Our Crime Lab consists of 10 different disciplines. They are question that I'm frequently asked is, how does your work compare to shows like CSI? Keep in mind, CSI was made for your entertainment, and crimes are not really solved in an hour. A major difference between a program like CSI and our lab is that on the TV show, the same person goes to the crime scene, works in the lab, investigates the case, interviews the victim or suspect, and makes the arrest. In our environment, those are jobs done by many different people. Our personnel in the lab do not interview any suspects or victims and certainly do not make arrests. The scientific disciplines require a 4-year degree in a hard science, such as chemistry, biochemistry, or biology. The DNA section requires very specific college-level coursework in genetics, biochemistry, and molecular biology. The breath alcohol, toxicology, and controlled substances sections require a strong chemistry background. The fingerprint unit requires an individual to be a certified latent print examiner. The investigative discipline does not require a specific degree.
Hi, my name is Brittany Baguley, and I work in the section that performs arson analysis. Arson occurs when a person deliberately sets a fire using an accelerant like gasoline or lighter fluid. The investigator collects fire debris from areas where they believe an accelerant was present. These samples are then submitted to the crime lab. We examine the debris to first determine if an accelerant is present. Even if only traces of the accelerant remain, less than 1/100th of a drop, there is a good chance that we will be able to detect it and possibly identify it as well. After fire investigators collect fire debris from potential arson scenes, they submit it to the crime lab for arson analysis. They'll submit the debris in airtight containers such as paint cans in order to prevent any accelerants that may be present from evaporating. When the can arrives into the lab, we'll open it briefly, note its contents, and then submit a charcoal strip into the airspace above the debris, and then reseal the can and place it into an oven to be heated overnight. The purpose of the heating step is to cause any accelerants that are present on the debris to evaporate into the airspace above the debris and be concentrated or absorbed onto the charcoal strip. After removing the can from the oven, we allow it to cool to room temperature. We then open the can, remove the charcoal strip, place the charcoal strip into a glass vial. In the next step, we use a chemical to remove any accelerants that may be absorbed to our charcoal strip. So this chemical rinses the strip, pulling off any accelerant that may be present. We give the vial a little shake so that all areas are rinsed, and then we transfer a portion of this liquid into a second vial and this is the vial that's placed onto the gas chromatograph mass spectrometer, or GCMS, for arson analysis. Most accelerants contain more than one chemical, and so we utilize the GCMS to separate and identify these different chemical components in accelerants. This process takes about 45 minutes.
Hi, I'm Kristine Albers, and I'm in charge of the breath alcohol section here at the Crime Lab. The breath alcohol program at the Washoe County Sheriff's Office is a state-funded program that is responsible for the calibration and maintenance of over 30 breath alcohol instruments in northern Nevada. I'm also responsible for training law enforcement officers in the proper use of these instruments. Operators of an evidential breath testing instrument are required to attend an initial 4-hour class followed by a 2-hour recertification course every 3 years. These instruments are placed in Reno and Carson City, as well as more rural locations throughout 13 counties in northern Nevada. They provide drunk driving or DUI offenders with a choice of either a breath or a blood test as required by Nevada law. We have instruments in place from Reno all the way to the Utah border, and we sometimes make road trips totaling over 1,000 miles in 3 days for calibration purposes. And now I'm going to demonstrate an evidential breath test on our Intoxilyzer 5000 model here. We've got the simulator that's attached to the instrument for the control for the test, and the breath tube here is where I'll be delivering my sample for the actual breath test. When an officer brings a subject to the breath testing room, the subject will most likely have a choice between either a breath sample or a blood test. Those of us who don't like needles will probably opt for the breath test. Once the breath test is chosen, the officer will then commence a 15-minute observation period, which is mandated by the state to make sure that the person does not put anything in their mouth that would affect the breath test, like alcoholic beverages or coins, things that people think may help their breath alcohol test results. So once that's completed, the officer will then initiate the Start Test button, at which point the Intoxilyzer 5000 will ask for a card to be inserted, which is the breath test printout card. Once that's taken into the instrument will ask the officer to input the start of observation time to make sure that 15 minutes have elapsed between the start of the breath test and the observation period. So I'm going to put in a number here. Then the instrument is going to sample some air from the room to make sure that there's no alcohol either in the room air and also flush the instrument out from any air that's within the instrument prior to my breath test. Then the instrument will test the simulator to ensure that the instrument can recognize and measure alcohol properly immediately prior to my breath test. And you can see here that the air is being forced into the solution and the vapor is being tested by the instrument. Now that we had a valid result for the simulator test, it read 0.102 grams per 210 liters like it should have. The instrument is continuing with the breath test. Another air blank prior to my sample that I'm going to deliver to the instrument. And then the instrument is requesting that the subject blow into it. Please blow into the mouthpiece. So I'm going to take a deep breath as the officer would have instructed me, and I'll deliver my sample real quick. Zeros, as it should be. An air blank to flush out my sample in case it was alcohol containing. And then the instrument's going to go into a 2-minute countdown and wait for my next sample. Our state mandates that we have 2 breath tests from a person within 2 minutes, and they need to agree within a 0.020 grams per 210 liters of breath. The 2-minute time period has elapsed, and now it's taking another air blank before it asks for my second sample. Requesting my sample, so I'll give that real quick. Again, my result is zeros. A last air blank to make sure that my last air sample was, or breath sample was blown through the machine and cleared out. Now the instrument's going to ask me for, or the officer actually, for some demographic information, the subject's last name, where the person was arrested, the citation number, etc. So I'll go ahead and type in some of those things now. Now the breath test is completed and it's printed out the results of the simulator test and my 2 breath tests, and printed out all the information that I typed in as the demographic information that the officer would have put in. And this copy is then signed by the officer and used in court purposes for proof of driving under the influence of alcohol.
My name is Diane Machen. I work at the Washoe County Sheriff's Office Forensic Science Division in the Controlled Substances section. In the Controlled Substances section, we analyze various substances looking for the presence of a controlled substance. We analyze things such as green leafy substance, white powders, or dark tar-like substance, again looking for something that is controlled under Nevada Administrative Code. To analyze materials for the presence of controlled substances, we'll use various types of tests. The first type of test that we'll use is what we'll call a screening test or a color test, where you add a certain chemical to your sample and a particular color reaction will tell you that you have a compound or a class of compounds potentially present. The second type of test that we will use is what is called a confirmatory test. For our confirmatory test, we use 3 different things. One is microcrystalline test. A microcrystalline test is where you add a heavy metal solution to your sample, and then you observe the crystalline precipitate through a microscope. And the crystalline precipitate that forms will tell you whether you do or don't have a particular substance present. And we mainly use these microcrystalline tests for methamphetamine identification. The other 2 types of confirmatory tests that we will use are gas chromatography with a mass selective detector, also known as GCMS. When you introduce your sample into this GCMS instrument, the first part of the instrument, which is the gas chromatograph, will separate your compounds. It can separate something like gasoline, which consists of 100 different compounds, into individual peaks. Then your compounds are introduced into the mass spec or mass spectrometer part of the instrument, where your compound is bombarded by a bunch of electrons, which causes it to be unstable and fragment. And that fragmentation pattern is used to identify your sample. And the last confirmatory technique we use is called Fourier transform infrared spectrophotometry, which is known in short as FTIR. You shine infrared light through your sample, and based upon the wavelengths at which your sample absorbs infrared light, you can then make an identification.
And I'm looking for the morphological characteristics like cystolithic bear claw shaped hairs. Only marijuana has those types of morphological features, looking for that bear claw cystolithic hair on the upper surface of the leaf and all the clothing or covering hairs on the bottom of the leaf. Now we're going to take it and put some of it in a test tube. Now we're going to move it to the hot plate over here. Just heat it a little bit. The heating just makes the reaction go a little faster. Now we're going to go back to the other side and add a little more chemical, a different one. The first chemical was the ducanoylevine. This is concentrated hydrochloric acid. We're going to go back and heat it. And with marijuana, you should see a purple color formation, which we are. So you're going to see my purple color. It's definitely purple. And that's the shade of purple you're looking for. And then we're going to add chloroform. This is the modified part of the test. That's why they call it the modified Duquesnoy-Levine. Now we're going to mix it up again, and that part of that purple color should actually go into the chloroform layer, which is on the bottom of the test tube. And so that's what you're actually looking for. You're looking for this color in the bottom layer, which is the chloroform, and then your aqueous layer is the top layer that should be purple. So that's a positive Duquesnoy-Levine test. So what we're going to do now is that hanging drop crystal test. And we just want a small piece. We don't want the big giganto chunks. That's good.
Okay.
And now we're going to move over here. And we're going to take a little cover slip and put a grease pencil mark on it. Only so our solutions don't run together. And then we're going to take a toothpick and some platinum bromide solution. And we just want a small drop, so that's why I'm using the toothpick. And we'll put it on one side of the grease pencil mark. And then we're going to take gold chloride solution and put it on the other side of the grease pencil mark, because we're doing two tests basically at one time. That's why we're using two different solutions. And then we take sodium hydroxide, which is a base, which is going to make our methamphetamine, if it's present, volatilize. Kind of mix it up with the other end of the toothpick. Invert our microscope slide. And now that sodium hydroxide is going to volatilize, basically convert the methamphetamine to methamphetamine base and volatilize it. And now we're just looking through for a crystalline precipitate if there is one, and there is. And the platinum bromide is positive for methamphetamine. And just look around the whole thing just to see what all the crystals are. And then we're going to take acetone. And the acetone actually doesn't dissolve the methamphetamine, it dissolves a common cutting agent known as dimethylsulfone, or DMSO2. So we're going to actually wash it with acetone a few times. So our methamphetamine's still in the tube, it's not dissolving, only the cutting agent would be going in. And we don't want any acetone in our tube because that's where the DMSO2, the cutting agent, will be. So actually just going to make sure I get all the liquid out of the tube. And now we're going to put chloroform through it, and the methamphetamine will come out in the chloroform. We're going to add just a little bit more. And just that rotating it just kind of gets the sample in contact with the solvent so that it
And the bottom
dissolves.
tube is a catch-all. You're using that for the liquid to drain out it, right?
Yep, the bottom tube, because I'm actually going to take this liquid now and test it. That first liquid was just a wash and a waste, basically. Now we're going to take and put a barcode on our tube so we can scan our barcode. So I'm going to turn on the hair dryer and then take my sample and spot it on the little window. And again, that hair dryer is just getting rid of the chloroform, and we're leaving our methamphetamine on our sample plate. I'm just turning up the light so I can see through the microscope so I can focus on my sample. And while it's doing that on the screen, you'll actually have an interferogram. It looks like methamphetamine. Now we're going to label our spectrum with our lab number and everything. And now I'm going to put my little barcode from my sample over at the barcode reader and scan my barcode as it is. And you're looking basically at wherever there's a peak here, there should be a peak here. And it found methamphetamine as my library match. And then we're going to print that.
Hi, I'm Dr. Lisa Smyth-Roam, and I'm a criminalist working in the DNA section of the crime lab. DNA is the fundamental building block of life found in almost every cell of the body. Here at the crime lab, we use DNA analysis to help determine if an individual's DNA profile was left at a crime scene. In forensics, we are interested in the 0.1% of the DNA molecule that makes us unique. A portion of this unique area of the DNA is used to generate a DNA profile. A DNA profile can be generated from cells deposited on items of evidence left behind at a crime scene. Such items of evidence might include blood stains, touched and handled items, cigarette butts, and even food. We can then compare the DNA profile obtained from the evidence to the DNA profile from a known individual, usually the victim and suspect. If the DNA profile from the evidence and a known sample are the same, we can then provide a determination of the statistical significance of the match.
This is our capillary electrophoresis machine. How this works is we inject our samples into these 4 tubes called an array. The samples then move through the tube at a rate that depends on the size. Larger fragments move slower than smaller fragments. Once the fragments get through our laser, then we detect the fluorescence, and this information is then sent to the computer and we generate an electrophoregram. For a full plate of samples, it takes approximately 24 hours to generate all the data using this machine.
Hi, my name is Jeff Riolo, and I'm a criminalist in the DNA section of the crime lab. My job involves managing the DNA database, or CODIS, which stands for Combined DNA Index System. CODIS is a computer database of DNA profiles from samples such as offenders and crime scenes. This system was established in the early 1990s by the FBI. The system started out with only a few states participating, but is now used by all the states and other countries as well. Since a DNA profile is basically a series of numbers, it is very easy for a computer to search and compare numbers. Offender samples are entered into CODIS if an individual has been convicted of a crime such as auto theft, burglary, robbery, sexual assault, homicide, or crime against a child. Where the DNA database becomes very useful is when a biological sample has been obtained from a crime scene. The DNA from this crime scene sample is entered into CODIS and compared to offender DNA profiles and other crime scene profiles at the local level of the Washoe Crime Lab database, and then at the state level, which incorporates the DNA profiles from the Las Vegas Metropolitan Police Department. Then the profiles are entered and searched at the National DNA Database, which the FBI manages. At the national level, all 50 states are contributing their profiles, thus our crime scene profile is compared to offender and crime scene profiles from across the country. This enables us to see if an offender from another state may have left their DNA profile at the crime scene.
Hello, I'm Ron Young with the Washoe County Sheriff's Office. I'm the Senior Latent Fingerprint Examiner for Washoe County, and I am certified in the field of forensic identification. I'm in charge of the WIN APIS program, which is the Western Identification Network Automated Fingerprint Identification Section. We've had 3 generations and changes through this system. Where it used to take 2 hours to search a fingerprint, we can now do that in 5 minutes with access to over 100 million records on file. The Western Identification Network is made up of the member states of Wyoming, Montana, Idaho, Utah, Oregon, and Nevada. Additional stations and states that were added were Alaska, Washington, and the Portland Police Bureau. We also have access to CalDOJ, which has been available since the inception of WIN on November 1st of 1988. The third generation includes the entire process of scan, entry, and search, which can now be completed in 5 minutes or less. It takes a long time because it downloads a list of up to 30 different possibilities. Okay. So what you do is you're looking at each one individually, and in this particular case, the very first one is an identification with this individual. So what we do then is we take this print and we look at it in this particular way, and I plot the points of the identification that was made by simply doing this. So then I can take this and make an actual copy of it on the copier, and we can put it in the folder with the identification with all the paperwork. And this gives us a copy of the identification already mapped out. And it has the SID number of the individual, where we can go into another database, which is called the Digital Archive System. And we take the number that the machine gave us, and then call it up on the screen. That's the individual that was identified, and this is the printout that came off of the regular printer. We also have access to the FBI Integrated Automated Fingerprint System, which is now available through the FBI. It effectively gives our agency the capability to search over 100 million prints.
Hi, I'm Kevin Lattyak, and I work in the Firearms and Tool Marks section. The Firearms section examines guns, ammunition, and ammunition components. We assist investigators by helping to identify weapons involved in shootings or stolen weapons with altered serial numbers. We also examine tools and items with defaced serial numbers. As examiners, we conduct tests on these items, and then we pass the results to the investigators. The final part of our participation in a case is testifying in court in front of a jury. Our task is to inform and educate the jury. The Firearms Tool Mark Unit examines a wide variety of firearms, components of fired ammunition, serial number restorations, gunshot residue pattern, distance determination analysis, integrated ballistic identification system, IBIS, and tool mark related evidence. Firearms are often used during the commission of a crime or with criminal intent where injury or death occurs. Evidence, particularly weapons, involved in these types of events is often submitted to the laboratory for function testing. This examination determines if an intentional alteration or defect exists in the manufacturer's operational design of a specific firearm. Function testing of a firearm is also performed to obtain test-fired exemplars, which are compared to fired bullets and cartridge cases that are often collected at a crime scene. The comparison microscope is the primary instrument used by the Firearms Toolmark Unit in the examination of comparisons of fired bullets and cartridge cases. This special type of microscope allows the examiner to compare the evidence components of fired ammunition to test shot exemplars simultaneously. Unique microscopic individual characteristics are imparted upon the components of fired ammunition during the firing process. These individual characteristics or marks are often referred to as stria and are unique to a specific firearm. This type of examination allows the examiner to identify components of fired ammunition as having been fired from a specific firearm. It is quite common to find firearms with obliterated or eradicated serial numbers. This practice of removing the serial number from a firearm is often the result of attempting to conceal ownership of a weapon that has been stolen. The Firearms and Toolmark Unit specializes and utilizes a chemical technique that is often successful in the restoration of a readable number. When a firearm is discharged at close proximity to an object, it deposits what is known as gunshot residue, or GSR. A distance determination examination establishes how far away the muzzle of a firearm was when it was fired at the victim. This examination consists of a series of microscopic and chemical tests, firing the suspect weapon at predetermined distances, and a final comparison between the evidence and tests to establish an approximate proximity of the firearm. The Integrated Ballistic Identification System, or IBIS, is a computerized digital imaging system which captures digital photographs of bullets and cartridge cases. These images are stored in a database and electronically compared to one another. An IBIS technician conducts a visual comparison of high-confidence hits or images that display similarities for possible identification purposes. This system can generate investigative information and connect firearms evidence from a previously non-related shooting events.
Well, we have two cartridge cases. The cartridge case that's on the left of the black line is a cartridge case from a drive-by shooting, and the cartridge case on the right-hand side of the black line is from a Reno drive-by shooting. And what you're looking at are the breech face markings that are left on the cartridge case after it's fired in the gun, and those markings would represent basically the bolt face, or where the cartridge case rests up against the gun when it's fired or discharged. And basically you're looking at it under a comparison microscope, so you're looking at two objects at the same time. We have one cartridge case again over on the left and one over on the right. And so the comparison microscope is really the workhorse of the unit that allows us to look at two bullets or two cartridge cases at the same time. Okay, these would represent guns that we've collected over the years. They've been submitted from various different agencies: Reno Police Department, Sparks Police Department, the NHP, and what they can do for us is allow us to look at the rifling impressions that are left on bullets and the tool marks that are left on cartridge cases for a particular type of gun. So we have tests on file, so we can go ahead and maybe use that information to tell an investigator when they have a shooting and they have a cartridge case or bullet but they don't have a gun, what type of gun it was fired from. So we can actually show the investigator the gun in question. We do serial number restoration here, where we're actually trying to restore serial numbers on guns that have been ground off or defaced. And so when we have a reference collection, we can actually go ahead and look at the same type of gun and look at the serial number and look at the size of the digits and the number of digits and letters in the, or characters in the serial number, and it can help us or give us assistance when we're restoring those obliterated or defaced serial numbers.
Do you ever have to fire any of these weapons?
Yes, we fire them routinely to collect evidence samples or to collect test samples for our file.
Are these weapons formally evidence,
It's my understanding that most of these guns were evidence at one time. If we have a shooting case where the gun isn't recovered, but we can tell the investigator what type of gun it was fired from, we can go ahead and pick out the same type of gun out of our reference collection and then go into court with it and show the judge and the jury and the attorneys what type of gun most likely fired the bullets that, you know, may have killed a person or been recovered from a crime scene. So having a similar type of gun is sometimes very instrumental in helping to solve crime and prosecute criminals.
Hi, I'm Suzanne Harmon, and I'm the supervising criminalist for the primary examination and DNA sections. The combined primary exam and DNA sections have replaced what was once called serology. By having these two sections together, the flow of evidence and subsequent analysis is more streamlined. The analysts assigned to the primary exam section are trained to locate and identify body fluid stains consisting of blood, semen, and saliva. Using chemical and immunological tests, specialized lighting, and microscopes, the analysts examine evidence such as swabs collected from victims and suspects and crime scenes, clothing, bedding, weapons, and vehicles as examples. The scientists in the primary exam section are also trained to locate and collect hairs that are on items, determine if the hairs are of animal or human origin, possibly identify body origin locations, and evaluate if the hair has a root with tissue attached that could be submitted for DNA testing.
In the primary examination section, we're locating and identifying biological stains. An example of that would be a possible or potential blood stain. Obviously, I want to find something that looks like blood first, so an obvious red, brown, or reddish stain. And in this case here, I do have a stain that is blood. A very quick presumptive test can be done using what we call phenolphthalein. So if I were to see a stain that I believe to be blood, perhaps on a shirt, and I would take a piece of filter paper and I can just scratch the surface of the stain with this clean filter paper. And what I'm doing here is I'm transferring some of the stain onto the filter paper so that I can do the testing not on the stain itself, but on the filter paper. So what I'm doing here is with my reagents here, which are colorless to start with, I'm going to place a drop on this transferred stain on there, and I'm also going to add a drop of some hydrogen peroxide. And in the presence of blood, it's being converted to a different compound, which is actually pink in color. And so you can see this immediate pink color, and this is a positive test then for the presumptive presence of blood. Now, human blood and animal blood would give this same reaction. But from this, I've now identified that this, what appeared to be blood stain, is in fact very likely blood. This stain then could be removed from the garment and forwarded then for subsequent DNA testing.
If we have belief that possibly there's a washed blood stain in a garment or in a carpet or something of that matter, we can use a chemical called luminol, which has the ability to react with non-visible blood. So what we would do in this instance, we have a shirt that we believe possibly had some blood stains that had been washed out. I would lay the garment out, and then working in the dark, I'm going to spray the entire garment with a light mist of my luminol reagent. It's going to react with the blood and produce its own light. We actually say it luminesces, and we would see kind of a glowing on the garment of the stain. While I'm working in the dark, any areas like that I were to see, I would take a marking pen and circle that area so that when I'm finished, I'll turn the lights on and then know where those possible stains were on the garment. And from there, I would proceed to further testing, possibly some more presumptive testing for blood, and even to cut the stain out and again send it to DNA. It's disappearing pretty quickly. Another type of test that we do is to help enhance possibility of seminal fluid on clothing. In this case here, there's a stain that's actually pretty visible to the naked eye of a probable seminal fluid stain. But in the instance that if this were a white shirt and I was unable to see the stain, I can use an ultraviolet light which will help show that stain up. And how that works is there's compounds in the seminal fluid that when an ultraviolet light hits it will give a fluorescence, and thereby helping me to see that stain. This is an example of a test that I would do to indicate saliva in a stain. And so looking here at the results of a test, you can see these clear circles on the blue background. Basically, I took a stain and mixed it with some water to extract the saliva into the stain, into the water. Put the water into wells that were cut into this agar. It's like a jello substance, and it diffused out into there. And we're looking for this clearing presence indicates to me that saliva is present.
Hi, I'm Karyl Brown, and I work in the toxicology section. Toxicology is the study of toxic substances and poisons. Forensic toxicology is the application of toxicology for the purposes of the law. The role of toxicology in law enforcement is to determine the presence and amounts of drugs and to provide interpretation and potential for impairment. In the case of the coroner and medical examiner, we act as a consultant for the medical examiner and provide information concerning roles drugs or poisons may have played in the individual's death. We're standing in front of the alcohol testing instrument in the forensic toxicology section of the crime lab. I wanted to share with you for a moment exactly how this instrument works. The instrument is a very pristine instrument, and we cannot inject blood sample directly on the instrument, so we have some preparation to do. Prior to explaining that, I wanted to let you know what kinds of testing is done here in the laboratory. We do ethanol or alcohol testing for a variety of different agencies, including law enforcement agencies for DUI, parole and probation, the Washoe County Coroner Medical Examiner's Office, drug-facilitated sexual assaults, and other types of testing in the northern Nevada area. Prior to placing the blood onto the instrument, we must process it in some form. The first step is to bring it to room temperature and to mix it gently. Once the blood sample has been mixed, a portion of the sample is pipetted, which is just a term to use to measure a small portion of blood into what's called a headspace vial. A portion of what's called internal standard, which is simply a tracer used for this instrument to make sure that the process works properly, is also placed into the tube. Again, this sample is mixed, and you can see that a portion of blood and internal standard looks like this. Each sample, each case is performed in duplicate, stoppered and crimped into the headspace vial, and placed on the instrument along with a series of calibrators and controls. These calibrators and controls help us to identify not only that the component we are looking at is truly the ethanol, but also how much ethanol is present. We have strict quality control criteria that must be met, and as long as all that criteria is met, all the cases on the run are accepted. If there is any problem with the quality control criteria, the run may not be accepted and must be rejected. Once the sample is placed onto the instrument, the system is ready to be started with a sequence that we've already prepared. The sequence simply tells the instrument which samples are to be run in which spot on the instrument, along with a series of calibrators and controls. The vials are placed on the instrument, and you will notice a turret, which is where the samples are brought into the system, heated and pressurized to what's called an equilibrium, and the area above the blood, which is called the headspace, is now to be tested. And since ethanol is a volatile compound, once it's heated and pressurized, it comes into the air above the sample, and that portion is actually what's inserted into the system for testing. The sample now enters the GC, or the gas chromatograph. It is a series of two separate columns. The samples go through both columns on the way through its analysis. Once the analysis is complete, values are generated, quality control acceptance is evaluated, and reports are generated. Those are sent to both the District Attorney's Office and law enforcement agencies. The Forensic Toxicology section of the Crime Lab also deals in drug testing. We perform drug testing on a variety of types of cases, including law enforcement, Washoe County Coroner and Medical Examiner cases, and parole and probation type cases. The first step of this process is a screening test known as immunoassay. It is performed by an instrument called the TCAN Mini Prep. This testing is performed, as we spoke with the alcohol testing, in a batch of samples, multiple cases performed on the same run with a series of controls. Once all the quality criteria are met, these results can be observed and retrieved. This technique is a screening technique known as immunoassay. It looks for classes of drugs. It is not run as a single sample, but in batches of samples, and these batches can take up to 8 hours to perform. Once results are obtained on these batches of samples, we consider them only presumptive screens. At this point, we rule out any case that is in negative or none detected on those screenings. If it's positive, a presumptive positive is not reported, but must be pursued through another technique known as an extraction process. This is the extraction area of the Forensic Toxicology section of the Crime Lab. This is where a lot of action occurs, similar to something like your old-fashioned high school chemistry lab. There's a lot of activity going on because we're trying to isolate the drugs that we identified previously in the screening technique and measure the amounts of drugs present. These drugs cannot be easily detected in a blood sample like this when we use our pristine instruments that we will show you in a moment. The initial blood sample appears like this, and our final result from the extraction procedure will be this very small clear fluid. The point of this extraction is to clean up the blood specimen and isolate the components of interest and concentrate them into a small solution in order to inject that on our mass spectrometers. These instruments are not set up to inject blood samples as a whole or urine samples and must have a more pristine sample. These extraction procedures can take anywhere from 6 to 8 hours each day. Once the extraction process is complete, the sample is then placed on the instrument known as a GCMS, or gas chromatograph mass spectrometer. We'll call it GCMS for short. The GC, which is this portion of the instrument, is the portion of the instrument that takes the sample through the system and separates the components according to their chemical and physical characteristics. Once the sample has come through the GC and into the mass spec, which is the second portion of the instrument, those components in the sample are actually exploded with a beam of electrons, and we get what's called a fragmentation pattern or a fingerprint of the molecule. And just like a human fingerprint, it is identical each time for the component of interest. These are then matched up with known standards and controls so that we can not only identify or confirm the drug that's present, but also measure the amount of drug and report that to the law enforcement agencies and district attorney's offices. This process is run in batches and takes overnight to complete.
My name is Marci Margritier, and I'm a forensic investigator in the Forensic Investigation section. There are 4 civilian and 5 commissioned investigators that work in this section. Our main function is crime scene processing. We respond to homicides, officer-involved shootings, sexual assaults, armed robberies, residential and commercial burglaries, and suspicious deaths. Crime scene processing includes photographing the scene, items of evidence, and any suspects, victims, and witnesses as needed. We routinely attend autopsies for photography and evidence collection. We also do shoe print, tire track, and tool mark castings as needed.
Hi, we're going to talk about the process of supergluing items for fingerprints. There are several different types of prints. One is referred to as a latent fingerprint, which means it is not visible to the naked eye, and it has to be processed with some type of chemical or powder to make it visible. So we're going to talk to you about supergluing, which is one of the primary types of processes that we use here in the lab. It is used on non-porous items, so anything basically that is not paper. What we will do is we will take an item, We will place it into a super glue chamber, which we have several available. The most commonly used is one of our fish tanks here. We do have larger ones in the background, and we do have some cylindrical types in another area of the lab here for the longer items. Anyway, we will place the item into the glue tank. We will then add, we have a heating element, which we would plug in here. We will take a aluminum tin. We would place that onto the heating element here. We also use a test slide each time that we process it to make sure that the super glue is actually working. So we have a test slide, and what we would do is actually go ahead and put our own fingerprints on here, our own prints on here, and then place it into the tank also. Then what we have to do is we have to apply, or I should say we have to add warm water, which we would place into a separate bowl here. Then we would place our item into the tank. We get the lid ready to go, and then we would then place our super glue actually into the little aluminum tray there. We would then close up the tank and let it actually cook for probably about 10 to 15 minutes until we notice that prints that are actually showing up on our slide here. Basically what it is that we're doing is we're heating up the super glue. The vapors then form and it sort of swirls into the tank. When we add the hot water, the vapor from the water also swirls in there. They mix together, and then it goes ahead and it lands onto the item that we're processing, and it would adhere to the residue left onto an item after you've touched it. After we noticed that we're actually getting prints on our test slide here, we will then go ahead and unplug the heating element. We would open the glue chamber, and then we would close the hood lid, so that way as it's venting, any of the remaining vapors that are in here would then be sucked out the hood. So that way it's not coming into the room, and it's not we're not breathing those things in, because that would be very bad for your lungs. Because super glue, what it does is it mixes with the water, so obviously your lungs have water in it, and you don't want to breathe that in. So once this has vented for about 10 or 15 minutes, then we can actually remove those items from the chamber and see if there are any fingerprints onto the item. We would take those into our powder processing room, and we will look at those with a loupe, and if there are any prints on there, we can either photograph it, or we can actually apply powder to it and then use lift tape and put it on a lift card to go to our latent section for comparison and analysis. The actual gluing process in the smaller chamber takes about 15 minutes. If we put it into the larger chambers, which are behind me here, these chambers generally take about at least 8 hours. Hi, now we're going to talk about processing porous items such as paper and cardboard with a chemical referred to as ninhydrin. What we do with this is we would saturate the item with the ninhydrin, and this is performed in a hood because we don't, again, want to breathe the vapors. So we just pipette a small amount out, and we go ahead and we get the item wet. Now on paper, because the ninhydrin would soak completely through the paper, just one application is fine, whereas on some of your heavier duty items, such as cardboard, you would want to go ahead and saturate both sides, but as you see, both sides are wet. Okay, and what we do with this item is then we would go ahead and hang it up here to dry. And it takes, for a paper like this, it takes approximately about 30 minutes for it to actually dry. After your items have been allowed to dry for at least 30 minutes, it's like with the super glue process, to where it takes the combination of the heat and moisture. It's the same thing is with the ninhydrin process. So to apply heat and moisture to that, one of the best ways to do it is with an actual steam iron, just your normal everyday household iron. What we're going to do is we're going to apply that to the paper. We don't actually touch the paper. We just hold it over the surface. And after applying the heat and the steam, you can see that the fingerprints that were on the paper are now visible. The process normally takes, for a regular type of paper, about 30 minutes for it to actually go ahead and dry. With cardboard, it would take a little bit longer because it is obviously thicker. Now, unlike the items that are processed with super glue, these prints cannot be lifted. So in turn, we would have to actually take a photograph of these prints to turn into the latent section for evaluation and comparison purposes, because we cannot submit the actual item itself. Now we can also process the sticky side of tape for fingerprints, because as you touch tape on the sticky side and you pull your fingers away, if on like your clear regular Scotch tape, you will notice that there are actually ridge detail there. So we can process the sticky side of those tapes to actually make those more visible. Okay, so what we would do is we can process it with several different types of chemicals. One of those would be referred to as gentian violet. One of them is actually referred to as sticky side powder, and the other one is actually wet wop. Now on your gentian violet, it is basically a liquid, and it is applied to the sticky side of tape. And basically what we do is we would take a brush and go ahead and apply it almost like paint very lightly to the sticky side of tape. It is left to sit probably for 15 to 30 minutes. And then what we do is we hold it under running water and actually gently rinse off the paint itself. And then what would be left, hopefully, would be your fingerprints. Okay. The other chemical, which is sticky side powder, is a powder, and it is mixed with liquid. And again, it is applied to the sticky side of tape. It is again allowed to sit for the 15 to 30 minutes and then is gently rinsed off. And then you would have the fingerprints. And the same thing with wet wop. It is also a powder. It is mixed with liquid, I believe that's water, and is again applied to the sticky side of tape. It is allowed to sit and then is gently rinsed off. Another item that we use is referred to as microsil. This is basically used for casting tool impressions for comparison to a tool that might have been used to cut a lock or break into an item during a burglary, but it's also used, we can use it for fingerprints. If we're having a difficult time obtaining a comparison print from a victim or a suspect, we can also use actual microsil and put it on the finger itself. It's allowed to dry until it sets up, probably about 5 to 10 minutes. Normally it sets up really quickly, and then it can just be peeled off. It is really similar to I would almost say like silly putty, but it's just a little bit thinner than that. But it is actually, it can set up and can be used for comparison purposes. For fingerprints, we would actually take a picture of the microsil impression and submit that to the latent section for comparison. Now another thing that we do out in the field in the FIS section is we will do castings of shoe and tire impressions because that are left in dirt. Sand, any type of soil. Because obviously we cannot bring that soil back for comparison purposes in the lab, so we have to do something to actually bring it back, okay? So what we would do is we use something that's basically like a dental stone. We have casting powder, and what we do is we add water to that and mix it up, and then we would pour it into the impression itself. And as you pour it in there, this is basically what it would look like poured into the impression itself. And the whole process from the time that we actually start mixing the powder until we can actually collect the stone itself is anywhere between 30 minutes to an hour. Okay, this is what the top part would look like. So once we remove that from the soil, hopefully we have a nice impression in there. And this in turn can be packaged as evidence and brought back to the lab for comparison to any victim suspect shoe, or if it's a tire impression, then we can actually compare the casting to those suspect vehicle tires or the victim vehicle tires. Now to go along with the impression evidence of the shoe casting, another type would be if the dusty print was left on an item itself, such as the print on the chair here. Now what we would do with this is, because it cannot be cast because it's not indented in anything, is what we can use is we use a gel lifter. It's basically like a big piece of tape that we're going to put over the top of the print itself. Okay, once the print is lifted, then what we do is we put the plastic protective covering back over the top of this to be able to submit it into evidence so it does not stick to our packaging. And then we will request for comparison to be made to any suspect shoes that we would have collected in the meantime. Again, with latent evidence, it is something that is not visible to the naked eye. So basically, like on the refrigerator here. If I go ahead and I touch the refrigerator and I move my hand, you basically cannot see the fingerprint there. So again, we would have to process it with something to make it visible. So another one of the processes that we have available to us is powder processing. Okay, basically, and it's really similar to a makeup brush. It's nice and soft and kind of poofy here. So basically, with a little bit of powder on the brush, what we want to do is we just basically kind of want to paint the area. And as you can see, as the powder is being applied, the print is starting to become visible. Now the next step, now that we have processed the item with powder, obviously we cannot bring the refrigerator back into the lab for comparison purposes for our latent examiner. So what we would do is we would apply a piece of tape to it, so that way we can lift the print and apply it to a lift card and bring that back to the lab for comparison purposes. So basically what you would do is you go ahead and stick the tape down, and you want to have it make really good contact with the surface. Okay, so rubbing on this after the tape is on there is not going to damage your print whatsoever. And basically, like I said, it's making good contact with the surface and burping out like any air bubbles that would be there. Okay. Once the tape is removed from the item, it is placed onto a latent lift card. On the back of the card, we have to fill out the proper information, which would be the agency case number, the address where we are, the item that it was lifted from, our initials and commission number. In the space on the bottom, what we can also do is we can also draw a generic picture of the item that we processed. And then we can go ahead and mark on that picture with an X the area that the latent was removed from. These are then submitted to the latent section for evaluation and comparison purposes. This process is normally used on your non-porous items. We can either do this prior to supergluing, or we can superglue the item and then process it with powder to lift it. In the field, this is mostly used on your larger items that would not be brought back to the lab for processing, or items that might be a collectible item that we would like to go ahead and leave there for the victim or the business owner. One of the primary duties as a forensic investigator is to document the crime scene with digital photography. The photos are used for investigative purposes for the detectives that would be handling the case, but it's also used in court to show the jury what evidence would be located at the crime scene, and to also show you an overall view of the area that we have processed. Another way that we would document the crime scene is by making a diagram.
basically what we would do is we would do a hand-drawn diagram of the room, the residence, or the scene itself. And then what we would use is we would either use a laser measurer or we would use an old-fashioned tape measure to actually make the measurements. Then what we would do is, if it is requested for courtroom purposes, is we could bring those measurements and the diagram back into the lab and put it into a computer program referred to as CAD zone to actually make a computer-generated diagram for courtroom purposes. The CAD zone is quite a lengthy process. I would say it depends on how involved your scene is and how many items of evidence that you have. If we're documenting the distance between, like, say, a gun and the wall of a room, or the distance between a gun and the victim, you have more measurements than you would if you're just diagramming the room itself. So it could take, I'd say, anywhere from like 4 hours on up to several days, depending on how many items of evidence that you want to put into that diagram.